Assalamu alaikum. I am Hafsawat from Department of Zoology, Wildlife and Fisheries from University of Agriculture, Faisalabad, Pakistan. And in this conference I am going to present on assessing histopathological alteration in black kites exposed to copper and lead contaminated environments with a focus on application for cellular viability and potential human health risks. First of all, we would like to explain what is black kite. It's a medium-sized raptor. The genus Milvus indicates kite, and species migrans means wandering or migrating, which is obtained from Latin word. We selected it because it's a good bioindicator due to its upper level or higher consumer level in the food chain, and it has a diversified diet which includes fish, poultry, leftover meats and garbage heaps. We can say that it's a opportunistic feeder also, and also it includes earthworm, insects and waste of slaughterhouse, small reptiles, amphibians, rodents and even birds. It can eat anything that you find it. What are the objectives? First, to calculate the contamination of lead and copper in black kites from rural and urban areas of Faisalabad. Basically, it's a comparison between rural and urban areas of Faisalabad. We selected Faisalabad because it's a textile industrial zone. We can say it's a hub of Pakistan industrial zone, and we compared the pollution level or contamination level of pollutants or heavy metals from rural and urban areas. Second is to check the heavy metal concentration in specific tissues. I selected liver and lungs of black kites to check the contamination level. Third is to identify or to check the effect of lead and copper on liver and lungs. What could be the possible diseases or what could be the possible impact on not only black kites but also in food chain, the other organisms, smaller or higher level, including humans. We selected two sites I already described, two sites, rural and urban. Rural area is included agricultural land, and urban zone is industrial zone. Also it's a higher level of traffic area. We selected 12 kites from rural area and we captured 12 kites from urban area by using bait trap or netting. We put meat and we just pour it with a chloroform, liquid chloroform, so that we can capture them. And after capturing, we digested the samples, and through atomic absorption spectrophotometry we quantified the concentration of lead and copper. The dissection was placed it was taken place in the biodiversity lab of Dr. Nazia Ehsan at Zoology Department, University of Agriculture, Faisalabad. We dissected black kites individually, separated the concerned organs, which were liver and lungs in my case, and I then dried the samples, which we just cut it off, and from oven I just dried them, chopped the organs in fine particles or pieces, then digested the sample. The procedure was we took 1 gram of liver and lung samples and placed them in the respective beaker, then added 10 milliliter of nitric acid, put it on a hot plate for 30 minutes and heated. After 30 minutes I added 5 milliliter hydrochloric acid and reheated for 15 minutes on hot plate, and then when thick transparent material was formed, I diluted it with distilled water and raise the volume up to 25 milliliter. For analysis, high-tech lab of University of Agriculture Faisalabad was selected, and we quantified and assessed the concentration of heavy metals there through atomic absorption spectrophotometry, and it was taken in milligrams per kilograms. For statistical analysis, we found significant results, like there was significant difference between rural and urban areas data. The data which was indicating the rural area was less contaminated than urban area. So results are here that site 2, which was urban area, had more amount of copper and lead, in both cases. So here is the mean concentration of heavy metals in lungs. When we compared the concentrations from both sides, you can see here site 1 has less amount of copper and lead as compared to site 2. So it means urban area has more polluted environment. Similarly, when we talk about the liver, in liver also both sides have a significant difference. Copper and lead has less amount at site 1, while at site 2 copper and lead is seen more in concentration. That is, there is a significant difference. It is 20.97 and is 31.77, and there is also seen the difference between liver and lungs, because liver is more toxicated than lungs. The effect which we have observed on liver was that there were cellular damage. Some kites has liver inflammation, some had abnormal growth of tissues or cells, which leads to cancer and also liver cirrhosis. And if we talk about lungs, there were also necrosis and pulmonary fibrosis. We saw that there is a respiratory damage, like respiratory disorder in some kites. So that was due to oxidative damage to lungs tissues. What could be the possible future directions? We can work on molecular biomarkers that are indicators for heavy metal toxicity in the environment in any metropolitan city, in any polluted area, or any area which you can understand that this can be polluted with some heavy metals, nanoparticles, or any kind of pollutants. Oxidative stress pathways can be described. We can work on immune response, genomic adaptations, and analysis of biochemical changes in living bodies, and these living bodies can also include human beings. And what is the impact on human health? Because raptors are at higher level and consumer level of food chain, so they are best bioindicator of the environment and what is going on in the atmosphere, what kind of pollutants are present in atmosphere. So we can describe it more accurately with the study of raptors, that the environment is suitable for living organisms or not, including humans. Because in Faisalabad, as we have reported many international level or at national level, that in Faisalabad most of the people has hepatitis, and hepatitis is due to polluted water, as it's a waterborne disease. So polluted water is also due to the industrialization. So this is all about our environment which is polluted, and we describe it in a certain way with the help of raptor studies, that is black kite. Thank you so much.